Okay, so welcome to the second part of this Bioinformatics online tutorial. I'm Dongxu. I'm a faculty at the Life Sciences Center and also ECS department at the University of Missouri. So I will talk about bioinformatics servers and the databases for studying protein. So this actually will be based on Dr. Moller's lectures. So he gave an excellent overview about some basic biology. So what I will focus on is the general protein bioinformatics tools. So these tools, of course, are highly useful and also in high demand. So if you look at the bioinformatics, there's just so many tools and databases. So I will show the portals. There are literally thousands of databases and many more servers. But a lot of it actually are not fully utilized. So there's a high demand in using bioinformatics tools. So that's why I like to give this lecture. For the bioinformatics researchers, it'd be good that you know some of the tools to do real data analysis, to study real proteins. If you can only develop some tools without knowing how to use the major tools for studying biology, it may not be that great, because just like a computer scientist, people may ask you helping some basic computer problems. If you say, I'm a theoretical researcher, I don't know anything about the real computers. Yeah, you could do that, but for the vast majority of people, it may not be that great, because very few people can really do highly theoretical work for career. So if you look at the bioinformatics area, majority of people actually, after graduation in PhD, they probably don't do work related to bioinformatics actually. They may use their skills to do general computing or other things. But for those who really utilize bioinformatics tools a lot, they tend to continue their career in bioinformatics. So that's why you can see using bioinformatics tools are really in high demand. So here, of course, we're mainly focused on protein-related tools. And then for general biology, you can see these tools are very easy to use. So you don't have to know programming. You can use these tools to study biology. And this tutorial is intended for general audience, including college students and also graduate students in biology not doing bioinformatics. So hopefully our lectures will be useful to you. And also in medical applications, so many cases you'll find a gene related to a disease or some mutations in the gene. You may want to know what kind of role this gene has in this disease. So having the tools we talked about can really help understand such questions. And then it's actually very interesting to study proteins. So protein, if you really see it, every protein has its own personality and it has its own story to tell in terms of evolution, in terms of its function in the biological system. So it's very exciting to study proteins. For myself, actually, I am not doing a lot of coding, but still from time to time, people give me proteins. I run all the tools, provide some annotation and information. In many cases, people feel my annotations are helpful. So it's actually pretty fun to do things like this. Here, we focus only on how to analyze a protein. The basic idea is that if I have a protein, only protein sequence, no other information, no gene expression information, no other isolate data for that protein, what can I do from protein sequence? So that's kind of the idea for my lecture. I will cover about 8 areas. So today I will give some introduction and also talk about a pairwise sequence comparison. And then basically I'll give 4 lectures. Each lecture will have 2 units. Then I will also cover this multiple sequence alignment and the phylogenetic analysis. And then we'll talk about this domain, superfamily, motif, things like that. And then next one will be protein function, protein interaction, and pathway. I'll also talk about the membrane protein analysis because membrane protein is somewhat special compared to globular protein. And then we'll talk about protein structure analysis and also protein structure prediction. So you can see these tools are very broad in terms of scope, and then they can really provide a very useful insight for the protein, and that can help to develop a useful hypothesis to study proteins. Many cases, when we work with experimentalists, we can help them to design experiments based on these predictions. So the typical questions from a biologist given the protein are the following. For example, they give me a sequence. They ask what kind of function this protein may have. Of course, in many cases, you know the general function, but the function we talked about is more specific. For example, it could be a kinase, but only knowing kinase itself is not that helpful. You really need to know what kind of a kinase, what is the substrate of this kinase, how this kinase is regulated by other proteins. So this kind of mechanism would be more useful for actual biology. And then, of course, people are interested in protein structure, interaction with other proteins, and some dynamics. So we're not talking about the dynamics in my lectures, but that's also very important. I just want to point out, you can run molecular dynamics simulations for this purpose. And then for protein structures, once you have an accurate structure, either determined experimentally or predicted by computational approach, then you can do a lot. You can do like mutation studies, energy changes, and also active sites, etc. Of course, people say nothing makes sense in biology without talking about evolution. It is pretty true in the context of protein. So when you see the evolution trace of the protein, then you know a lot about its function. So many times we study this so-called phylogenetic footprinting. That means for protein, from the evolution in terms of a more ancient species to the newer species, what is the footprint in the evolution? Where it starts to diverge, what kind of function it gains through evolution, and what component is conserved. So all those things are very helpful to understand the protein. And then, of course, the disease and the phenotype. So in the case of a disease, many cases you find somatic mutations. Then you want to know what kind of defects this mutation can cause on this protein, whether it's related to disease, and whether it is a good marker for the disease, whether it is a good therapeutic target for the disease. All these things start from proteins. And then in agriculture, we often study the phenotype, like a certain trait for protein content, oil content. Maybe we can use a GWAS or genetic selection to find target proteins. But then what roles these proteins play in the trait, it'll be useful to use protein sequence to do such analysis. So there are many tools and database for studying proteins. Yeah, I would just focus on the tools for beginners. So they are all web-based, easy to use and understand. But even though they are simple, they can really cover a lot of use cases. I would say even majority of use cases, you can focus on a handful of tools and databases. Yeah, if you want to do more in-depth analysis, you need to look at the minority tools. And also you may need to use more advanced tools to get more accurate results. But a lot of cases, you don't have to do that. So these tools are certainly popular. More importantly, they are sustainable, because bioinformatics is more like small business. Majority of tools cannot sustain beyond 2 years. So people actually did a study a few years ago. They found I think like 80% of the servers, databases related to bioinformatics are no longer available after 2 years. So sustainability is actually a major problem. But fortunately, major tools, particularly the tools that I talked about, they will be sustainable for a long time. And then the question is where to find these tools. Of course, we'll talk about some most basic tools. But if you have a special problem, or if you want to use a more advanced method, how to find out these tools. So here are some ways to search for these tools. First of all, you can simply search for Google. So Google, the advantage is pretty up to date. For example, you have some latest tool, Google can provide the information. And then ChatGPT is actually a very good source to provide information. And then there's a relatively high quality tools. You actually can focus on this nucleic acid research database, server issue, this or web server issue. These tools tend to be in high quality because they have a very good quality control. We have a few tools published there, and they really did a thorough work to do evaluation, all kind of testing. So these tools are typically high priority in terms of choices. And then there are some other bioinformatics resource portal. There are quite many actually. My lab, at a certain point, we actually maintained the portal, but we just didn't have the manpower to keep up with it, so we didn't continue. But these two tends to be the most comprehensive. One is this database commons. One is bio tools. So I'm going to demonstrate these tools so you will see what they have. Okay, and then for this gene and protein database, we typically talk about the 3 types of databases. One is so-called primary databases. So these databases host experimental data directly. They're all very reliable, no computational predicted data. So related to gene and the proteins, there are 3 major ones. They have been dominating the field for at least 20 years. So the first one, called the GenBank, it's an NCBI's resource. It contains all the genome, gene, protein information. And then the AMBO one from Europe is also a good resource for protein and gene data. And then another one is from Japan, is called DDBJ. So these are the 3 major ones that you can focus on. And then there are a lot of so-called secondary databases. So they host the data from computational analysis on the primary database, including some annotation. For example, the interpro for protein families, motifs, and domains. And then Ensemble is for genes, variation, function, regulation. So they are very useful because even though you can run tools yourself to study these proteins, but it's a lot easier just to look up from the database. And then there are some databases we call the composite databases. That means it's a warehouse of many sources, many types of databases. For example, the UniPro we're going to talk about, they have a lot of information related to the protein. And then another big category, we call it the species-specific databases. For example, this is a MGI for mouse, fly-based for Drosophila, warm base for C. elegans, Tier for Arabidopsis, and then Soy KB actually developed here for soybean. So these domain-specific databases provide rich information for genes related to that particular species. So we're going to have a short demo of a Tier. And then just want to talk about this UniPro. UniPro actually stands for Universal Protein Resource. It actually has a long history. Initially, it was started from this Swiss Pro in 1980s. And then it has some expert annotation, and then has been maintained by this Swiss Institute of Bioinformatics and the European Bioinformatics Institute for many years. And then the quality becomes better and better. The information becomes richer and richer. But that only covers well annotated proteins. Another source actually came from the PIR. That's actually started from this Georgetown University. So it is a big collection of many proteins, much more than Swiss Pro. But annotation is not as rich as Swiss Pro. And then in 2002, these two sources are merged together with actually some other smaller databases to become UniPro. And then here is an example I want to highlight. This is the so-called model organism database for model plant Arabidopsis. As we know, that's really the most widely studied plant species by the research community. And then it has all kinds of information from sequence, genetics, physics map, gene expression, protein, mutation, etc. And it was started in 1999, but interestingly, it actually cannot sustain itself by this free software. So initially, they were supported by National Science Foundation, but later, National Science Foundation stopped supporting this resource, and then they have to use a fee-based subscription model, and currently run by this non-profit organization. It's called Phoenix Bioinformatics. So next, I'm going to do a few demos about Google Search, ChatGPT, and database common, BioTools, UniPro, and Tier. Yeah, I just want to mention that this tutorial is for education purpose only. So it contains some online material from public domain. I just want to acknowledge, I may not explicitly put the source on every slide, but just want to have a general acknowledgement.